right, our second part in our Meiosis unit on mitosis in the cell cycle, we'll be starting learning target 2. Students will be able to model the stages of mitosis and explain why mitosis is important to multicellular organisms and cell differentiation. Our objectives for this PowerPoint are, let me get my pen working here, to be able to list and describe the 4 stages of mitosis and cytokinesis, model the stages of mitosis, that will be an in-class activity that we do, and identify the stages of interphase and mitosis in onion cells using a microscope. That will also be an in-class lab that we do. With mitosis, the first phase is prophase. This is where chromosomes shorten and thicken. Okay. Remember that during interphase, the DNA was copied. That means the chromosomes were copied because they are made up of DNA. So you went from a single chromosome like this to a replicated chromosome like this. Now you have 2 chromatids held together by a centromere. Okay, that's what a replicated chromosome is. The nucleolus and nuclear membrane disappear, and the centrioles move to the opposite side of the cell. Those are going to be important for where the spindle fibers will grow out of, okay? And that's what's going to attach the chromosomes. You can't really see them on these pictures, but this right here is an animal cell, and this right here is a plant cell. This is what your mitosis onion root tip cells will look like. You can see that the nuclear membrane is starting to break down, and you can start to see it's not just chromatin anymore. You can start to see little thick little chromosomes in here that are thickening. Same with here. It almost looks like thick worms, but they are starting to form chromosomes here because they're really winding up tightly. The DNA is just winding up really tightly to form those chromosomes so that they're becoming more visible. But it's not really a tight circle anymore. See how this is kind of like folding in just a little bit? It's because the nuclear membrane is breaking down. The second phase of mitosis is metaphase. Meta means middle. Okay? The chromosomes are going to line up in the middle of the cell, and the spindle fibers will attach to the centromeres. So here are those centrioles, and that's where the spindle fibers are going. You can very faintly see these little lines here. Those are the spindle fibers, and they're going to attach. And these chromosomes, you can see, line up straight in the middle. Here they are again over here, and you can see these faint little lines. Those are the spindle fibers attaching to those chromosomes. But whenever you see chromosomes lined up in the middle, meta means middle, so that would be a cell that's in metaphase. The next phase is anaphase. This is when the spindle fibers start to shorten, and as they shorten, the chromatids get pulled to the opposite sides of the cell. So they actually get separated. You can see them here getting separated to the opposite sides. It almost looks like some spider legs or something, but those are the chromatids getting pulled to the opposite sides of the cell. And you can see that little gap. So they're no longer lined up in the middle, they're actually pulling apart. And finally we have telophase. Telophase is when chromosomes are at the opposite sides of the cell. The nuclear membrane and nucleolus reform. And here's where you'll start to notice a difference between animal cells and plant cells. In an animal cell, the cell membrane starts to pinch in. Okay? You can kind of see it right here. And in a plant cell, a cell plate starts to form. That's this little line right here. Okay? And you can see that the chromosomes are on opposite sides. So once you start to see that cell plates start to form in a plant cell, or that cytoplasm the cell membranes start to furrow or pinch in, then you know that cell is in telophase. It's not totally split yet, but the nuclear contents have split. That's why we're at the end of mitosis, because mitosis is the splitting of the nucleus, or the splitting of the nuclear contents. We haven't split the cell yet. That's what happens in cytokinesis. This is the division of the cytoplasm. So the cell will pinch into two in animal cells, and the cell plate will divide the cell into two in plant cells. So here's the cell plate now. Remember, it started out just right here in the middle in telophase. Now it has grown all the way across and divided the cell into two. Here, it started to furrow in just a little bit during telophase, but now you can see a line has come all the way here, and it has pinched, and these two have now split into two separate cells in animal cells. So that's cytokinesis, the division of the cytoplasm. So a little story I like to tell, a way you can remember all of the stages of the cell cycle is just to remember the story of the nervous wrestler. Have you not heard that before? Well, we have had plenty of state champion wrestlers in our school. And one of them, when he stepped out onto the mat, I mean, he had won all of his matches. He was a state champion. And so when it was his senior year, and it was his final match, and he was stepping out onto that mat for his championship wrestling match, the poor guy who was opposite him that had to wrestle him, he stepped out on the mat, and he looked up, and he took one look at him, and he basically looked at him and said, "I pee Matt. See?" You know, he's so nervous. Get it? I pee Matt. See? Well, that's just kind of a corny acronym for you to remember. Interphase. That's the first stage of the cell cycle. And then we have prophase, metaphase, anaphase, telophase, and cytokinesis. Remember, these four phases right here are mitosis. Okay? So if you can never remember all the stages of the cell cycle, you just have to remember the nervous wrestler. I, P, Matt, C. All right? Yeah, you're only going to get corny jokes from me. Sorry. All right, let's do a little practice. Now, I want you just, in this case, to see if you can find the four phases of mitosis as well as interphase in these pictures. So this one here, it shows that the cell plate is starting to form. Do you remember which phase that was? That was telophase. It's not quite split into two yet. Cytokinesis hasn't totally happened, but the chromosomes are on opposite sides and the cell plate is forming. This one here, you can see it looks kind of grainy, so I can't really see chromosomes yet. And we have this nice circle here, as well as this nucleolus. So this would be interphase, because the nuclear membrane and the nucleolus are still there, but the chromosomes are not visible. They're still in the form of chromatin, so that would be interphase. Here, those look like little, I don't know, they look kind of like tarantula spiders being pulled apart from each other. The chromatids are being pulled to opposite sides or away from each other. So when I think of away, I think of anaphase. Okay. Here the chromosomes are lined up right smack dab in the middle. Well, remember I told you that meta means middle, so that would be metaphase. And this last one, oh, I can see chromosomes. Look at, they're nice and thick. You can see them in here, but I don't see a nice tight circle. This looks pretty loose here. That means the nuclear membrane probably broke down, so that would be prophase. Okay, so you're going to have to find those when you do your onion root tip lab under the microscope. You're going to have to find these different phases and be able to identify them. And that's the end of this section.